I think we're ready to get started. So thank you all for participating in this coffee seminar. And as appropriate, we'll begin by an acknowledgement of country. And so we acknowledge the traditional owners and their custodianship of the lands on which we meet today, and pay respects to their ancestors and their descendants. So just a little bit of housekeeping before we get started. Christine will give us a presentation which will run for somewhere between 30-35 minutes. And as Christine is giving that presentation, at the bottom of the video that you're all viewing, you'll see the question and answer tab at the bottom of the screen where you'll be able to actually enter questions. And then at the end of the presentation, I will be able to direct these questions to Christine. And so then Christine will be able to answer, and depending on how many questions we have, we'll try and get through all the questions that are actually lodged. So with that, it's my great pleasure to actually be the person to introduce Christine. And just for those of you who are less aware of Christine's background, Christine's a professor of plant physiology in the School of Biological Sciences at the University of Queensland, and also a QAAFI affiliate professor. Christine, throughout her career, her research has focused on developing and understanding of the physiological and genetic mechanisms underlying traits, and using the tools of molecular biology to study whole plant development. Christine, with her career, is recognized as a fellow of the Australian Academy of Science. She is a highly cited researcher and has been a past president of the International Plant Growth Substances Association. Christine is currently an Australian Research Council Georgina Sweet Laureate Fellow. And Christine is well known for her groundbreaking research on the roles of strigolactones and sugars in the signaling networks involved in plant branching and plant development. That's not going to be the topic of Christine's talk today. We're fortunate today that Christine is going to give us an overview of the new Australian Research Council Centre of Excellence for Plant Success in Nature and Agriculture. So with that introduction, it's my great pleasure to hand over to Christine. So Christine, if you can, your video, and you should be in control.
Thank you very much, Mark, and thanks for that, perhaps that long introduction. I appreciate you going to the trouble to say all that. Yes, so I'd like to share with everybody the excitement and future ideas that we have about this new Centre of Excellence for Plant Success in Nature and Agriculture. So I'll start off by just sort of talking about what do we mean by plant success. And really, I sort of say to people, it is what you want it to be in a way. But really, when one thinks about plants, it's not just about the food we eat. We think about fuel, fibre, wood, chemicals for pharmaceutical purposes, and many diverse purposes. And of course, it's one of the obviously most successful forms of life on this planet. And so we as plant scientists, we really have a role, an important role to play in ensuring the success of plants, because it really means the success of the humankind, and also the success of other diverse organisms which depend on plants. So from the point of view of putting a bid into the Australian Research Council to make sure that plants are figured in the strategic direction of government funding, it's common to talk about this major problem here, which is the fact that the world population is dramatically increasing, and in our lifetimes it's going to be continuing to increase, such that it's going to reach just under 10 billion, about 9.7 billion people, we think. And this is going to put a downward pressure on the amount of arable land. And that obviously has huge consequences for our planet. The other problem is, of course, that, how are we going to feed the populations, and how are we going to do this sustainably? So plant breeders for all of this century, more or less, have been really successful in getting about a 1% genetic gain per annum in terms of improving crop productivity, so that we can use the same amount of land to get more product. And what the problem is that we're facing is that in order to match this increasing population, we need to be looking at doubling that rate of genetic gain. And at the same time, with climate change, it's putting a downward pressure on productivity of crops, where crops are greatly affected by heat stress. So with heat stress in mind and global increasing populations, we need to be very effective as a group of plant scientists in delivering this food for the human race and all of its needs, as well as the sustainability for our planet. And how effective have we been so far? Well, I would say extremely effective. As I said, that 1% increase in genetic gain. And this has been delivered in three main crops. And if you're off to dinner with somebody tonight, just who isn't a plant scientist, I think it's quite interesting table conversation to recognise that actually 2/3 of the world's calorie intake, the humankind's calorie intake, comes from just these three crops, which I think is quite amazing. That's an amazing feat for the people who've generated that outcome, but it's also quite interesting and impactful for our environment. At the same time, we can recognise that 10% of the world's population is undernourished, despite having plenty of food, plenty of this sort of food. In some cases, and of course in other cases, they don't even have that. Now, so that's a need. We've got to think about diversifying and improving crops. Why can't we do it faster than a 1% every year? One of the biggest problems is that plants are incredibly responsive to environment. So the amount of resources that huge companies have to put in to try and manage the crop improvement is absolutely immense. And, across many seasons, across each dot here represents plants or fields rather of plants that have been grown right across the US, in the case of the Aquamax breeding program that in fact Mark Cooper was leading, and looking at drought and well-watered conditions. That's a scale that we need to be thinking about to improve plants. So it's not really a great surprise that we've focused on a small number of plants for crop improvement. But this poor predictability means that these genetic cones are incredibly costly and inefficient, and so we need to be able to solve that problem. We also need to be able to predict in a new environment, such as hotter or drier environments, and we want to be able to think about new combinations of genes that we've never utilised before. Now, one of the other problems that we face is that all of the sort of fundamental discoveries of plants are studied in isolation. So what I mean by that is, developmental biologists will work on one aspect of the plant and not another. A molecular biologist might focus on just one gene. An ecologist might focus on one environment or one particular question. So that makes it difficult to think about the whole plant as a system. And it's very, very difficult to extrapolate from these well understood plants into a new set of plants. I'm just giving here the QAAFI example that I'm familiar with Hort Innovation, and that is of mango, avocado and macadamia. I mean, how are we supposed to get knowledge from these model systems across to these plants? It's going to be really difficult to do that, and we need to build the tools to enable it. So our vision is to accelerate our ability to predict and improve plant performance in diverse environments. And if we could do that, then that will enable the sustainable and productive future of plants in nature and agriculture. So I've highlighted in bold green there the ability to predict and improve. So prediction is a major focus of what we wish to do. So with this in mind, we want to be able to build a team that can deliver this. So by design, we've created a team with that capacity, and that team has come up with the vision that I have just presented of accelerating our ability to predict and improve plant performance in diverse environments. And we can achieve this through linking mechanisms and then putting those linked together mechanisms through models and then innovating the path to crops. So I'll go through, first of all, the team, and then exactly what we mean by doing those things. So first of all, I'll talk about the people that work on mechanisms. So we have myself working on shoot branching, Tim Broadweb working on water relations, Eloise Fu on symbiosis and the uptake of nutrients from the soil, the connectivity of processes to each other through plant hormones and photostimulates is Stephen Smith, and working on flowering, Jim Weller. So together that group covers much of the aspects that underpin plant success and plant fitness. And to view that from a more ecological perspective, we have the strengths of Ian Wright and Daniel Ortiz-Barrientos. And then John Bowman has a technology to sort of link these things together by working on basal plants and thinking about the core basis from which plants have evolved. So in order to do, to connect those, that thinking together, we need some network thinking and some capacity to build models that can connect the mechanisms to each other. And so we have Diane Donovan working on network theory, Graham Hammer, who many of you know, of course, from QAAFI, working on modelling crops, and Mark Cooper looking at quantitative genetics and big data. Kevin Burridge is also at QET and working on stochastic modelling, also have a long history in computational biology. And in order to predict out into diverse plants, we have Barbara Holland working on phylogenetics. And then in terms of the path to crops, of course, QAAFI former director Rob Henry, in the pre-breeding space, Dave Jordan, Peter Waterhouse working on network gene technology, CRISPR technologies, and Brad Sherman working on intellectual property and the law. So I just want to introduce those people because they're the people who've actually come together to build this vision for the centre and to create the path, we hope, to plant success. Just to finish that off though, I've just highlighted in purple there the people who are actually located at the University of Queensland, in red, the University of Tasmania. Then we have, as I mentioned, QT, Kevin Burridge and Peter Waterhouse, and then Ian Wright and John Bowman at Macquarie and Monash. So in order to achieve this, we have formalised partnership relationships with the universities I've just mentioned, as well as our industry partners and collaborators at other universities. And we have designed a program that will be delivered over 7 years. Now, we will certainly have ongoing linkages with people that we haven't yet formally developed contractual relationships with. But I'd like to say, take a moment to say that we really intend to be inclusive with the Centre for Plant Success in Nature and Agriculture for the reason that it is a big question and that it's about helping the whole of plant science community think about how to be inclusive to solve these really big problems in biology. So one of our aims is actually to enable success in others. And that is something we will be discussing internally and externally throughout the life of the centre. So what do we actually plan to achieve? We want to create a step change for plant success. And when I talk about that, I'm really discussing, you know, instead of a 1% genetic gain, we're talking a 2% genetic gain, that kind of step change. Doesn't mean we're going to solve all the problems of plant biology in 7 years, but we have some quite specific outcomes in mind. So our program rather, can be thought of across those three aspects I mentioned earlier: mechanisms, models, and path to crops. So our first theme is to discover and integrate different mechanisms of plant success in different environments. So that's taking, in large part, prior knowledge of different mechanisms and using that to actually combine them together and create new knowledge from understanding those interactions, as well as an ability to use that knowledge for prediction. And so that's where the modelling comes in. Develop models that connect mechanisms and genomes for predictions. So this is very explicit in that it's trying to link the mechanistic understanding from gene to phenotype. And the way to do that is through building predictive models. That will improve our genomic prediction, but it'll also be used to then test our understanding. And then the third aspect is innovating the path to crops, including gene technology, law, and society. We have many opportunities with gene technology and so on. So I've just sort of squished that image down a little bit and listed two sort of maybe simpler sentences that explain what we're trying to achieve here. The first is that we wish to improve understanding of plants through integration of knowledge and prediction. By predicting using existing knowledge, we understand what we don't really understand. But we can also use that to make better decisions or to improve our decisions in agriculture, in conservation. And that really is about innovating the path to crops and innovating the concepts of conservation through building capacity to predict and translate. So building the capacity to predict and translate is really about building the models and then delivering the path to crops. So it's obviously an incredibly diverse group that I'm presenting to you, but let me enable a little bit of focus on what we're actually talking about here. So in the first program, we're talking about integrating networks. And so one way to think about that is, you know, is the level of focus that we've currently been bringing to our disciplines and trying to bridge those disciplines. So I'm just describing, if you know, excuse my naive interpretation here, because everyone in a discipline will have a different definition, and that's absolutely fine. I'm just trying to kind of give you a sense of different perspectives on things. So for example, ecophysiology. An ecophysiologist would think of drivers of success or fitness as being multiple interconnected physiological processes and traits. So that's a layer of thinking that they bring in to the question. Developmental biologists will focus perhaps more on genes and on very specific processes that go on, particularly signals and, you know, transcription factors and all these sort of things, many of which are turning processes on or off, and sometimes regulating processes in a quantitative manner. A crop biologist would think of the same thing in the lens of resources and competition. So the size of a leaf will have implications for the next few days worth of photosynthesis and how much sink strength is involved. And so there's a lot of source-sink relationships, which is overlaying some kind of reproductive strategy or genetics. And then there's the breeding perspective, where, or even thinking of the natural systems, where we try to look at populations and genetic effects. And the bulk of thinking there is to think of genetic effects as being additive, which is the infinitesimal model that Mark Cooper's been teaching me about since we started talking about this. So you can see different perspectives here on really the same thing. So part of our goal is, in our step changes, is that we aspire to, the step change we aspire to relies on our ability to integrate and connect networks that underpin the above. So to be truly successful, we need to think, well, what are the technologies that are actually going to enable us to connect and integrate the networks that are discussed by these researchers? Let's build that technology, let's utilize it, let's test it. So this is the sort of findings that that physiological mechanisms across diverse plants can deliver for plant science. So this is work of Ian Wright, and he's worked on 2,000 different species, not 2,000 plants from the same species, but 2,000 diverse species, and looking at them in different habitats and then measuring traits. You can see a really clear trade-off between nitrogen and water use efficiency in either drought adapted or low nutrient adapted plants. So you could think of this as a kind of rule of success in plants, depending on, which is a sort of identifying trade-offs within plants. Another perspective on physiological mechanisms is coming from Tim Brodribb's group, where, for example, here, he's looking at how leaf vein density limits photosynthetic capacity, or descriptions of plant cavitation. So at what point when plants become water deprived under drought, do they actually lose their ability to take up water due to plant cavitation? So there's sort of fundamental laws of physics, if you like, which can be derived from these types of approaches, and which can be taken across to the sort of molecular domain to understand the genetic basis. Coming from the other direction, from developmental genetics, where we typically start with phenotypes from mutants or transgenic plants, and then we use these to dissect different components of a system. And this is the branching system that we've been working on for some time. And we could think of many different genes that are underlying these compounds, plant hormones, strigolactone, sucrose, and auxin, and so forth. And we can look at how environment affects these levels of these components and the phenotype that we are measuring at the end. And of course, overlaid on this is the computational models that can be built by people like Graham that look at how the plant has excess sucrose or very limited sucrose at different developmental stages, and depending on the environment and relative rates of leaf growth and so forth. And so if we can overlay that with these models, we can get to a much better predictive capacity. So this is sort of model which Graham would utilise. This is an example from Karen Chenu, and it tries to capture the major processes that are regulating the phenotype of crops. And so within the centre, we hope to take models such as I've described before, which are descriptive models based on molecular genetics. Here's one for branching, another one for flowering, and try and combine that with the kinds of models which Graham and others would produce. And then once these models have been combined, we can make much better predictions, we believe. So the current approach, which is used for predicting the gene to phenotype continuum, we would have on one hand an infinitesimal model here where the genes are all additive, or maybe there's some epistasis leading to the yield. And then a current technology incorporates kind of Graham's models, which are physiological models without genetics, but which we can pin particular genes to be underlying different processes. What the centre would like to do is to incorporate very explicitly current knowledge of genetic networks and how they actually feed into these processes. And that would enable a more explicit gene to phenotype model. So what plants have we chosen to do this within? Well, we'd like to use Arabidopsis as a model system simply because it's very tractable. Also, Daniel Ortiz Barrientos has a very nice natural system that we can utilise, which he's already done an incredible amount of population genetics work and has a focus on trying to understand the basis of adaptation. And of course, within QAAFI, we have a very strong sorghum group. So that's not our entire list of species we'd like to work on, but when we have focused programs that are aimed at building these particular capacities that we need, we will be identifying the best system to do that within our capacity. So the goal, it's partly to understand how to improve sorghum, or how to discover new branching genes in Arabidopsis. But really, this prediction toolkit's where we're really at. It's the maths end of things, building the capacity to link mechanisms and build predictions through biological modelling. If we could do that, then we've opened up huge opportunities for the future, whether it be just simply understanding biology or using it to improve crops or enhance sustainability. So I mentioned earlier about this difficulty to extrapolate beyond the species of focus. So I'm just switching gears just for a moment to think about, well, how would we solve that kind of problem? So far, I've been talking about, fairly straight crop growth model or crop pipeline. But what about, these sort of more difficult to work with crops where there's far less information about the physiology and mechanisms? What we'd really like to do is to be able to look out across what is known in the diversity of plants and say, for a particular chosen crop, is it more like this, that, or this other crop? How do we link as much as possible prior knowledge to enhance our understanding of these other crops? So one way to do that is to think about phylogenetic network profiling. So where we have prior knowledge of a network, let's say this is a branching network, this is the flowering network, and this is some other network. And this is how they all interact with each other and within those networks. This is the prior knowledge. And of course, there are a large number of these networks for branching or for flowering that are a little bit different in different species. And we already have that prior knowledge. But just imagine at the moment that we're just going with a blue network, a red network, and a green network, which we're saying it might exist in a single plant. What we now would like to do is work out how that network has changed. How does it differ across different species? So we have species A, species B, species C, and for each of these three networks, the blue, the red, or the green, we can look at how it's changed in the different species. And so what we already know the sort of phylogenetic history of those species through a huge amount of knowledge that's out there in terms of the phylogenetic origins and evolution of the species. So if we sort of fix that tree in terms of its structure, and then we look at how the different networks have evolved in each case. So this is the green network, the red network, and the blue network. And you can see how the red network, for example, in species B has changed more over time than it has in species A. So that means that network has diverged a little bit more than expected, given its phylogenetic background. So what we hope to do once we've accumulated this kind of data for our networks across diverse plants is then to build a matrix which describes these differences and then be able to plot that sort of data in a three-dimensional space to be able to, if you like, cluster species together in terms of a particular network. And we hope then to be able to infer a greater amount of knowledge by the association of that network with similar networks in diverse species. So that's just the very beginning of a kernel of an idea there. That's the approach that we're going to start off with, in thinking about how to look at the evolution of networks so that we can infer more information about species that we may not currently understand much at all about. So in our prediction toolkit, we're hoping to add a new idea of network phylogenomics as an outcome of the centre. Okay, so just summarising the first two themes that I've just presented, which are, just over 2/3 of the centre's energy. So I'll just summarise that for you now. We aim to link networks to each other to predict and understand how changes in one part of a network may affect another. That's really important, because if you're studying something in isolation, you cannot achieve that. If, on the other hand, you're able to link networks together, then you can answer those sorts of questions, and then you can have a much greater predictive capacity of plants as a whole system. And of course, that's what we need to achieve. That sort of ability, that network knowledge, will enable us to reduce uncertainty in genomic prediction and improve the gene to phenotype trait modelling. So whereas up here, I'm talking about enhancing understanding. So I'm going to learn more about branching by understanding how flowering and water relations impact on branching. That's understanding. But the goal here is to use that knowledge, in addition to that understanding, to reduce the uncertainty in genomic prediction so that we can improve the gene to phenotype trait modelling so that we can have direct outcomes for crops. We need to build that technology and then apply it in quite focused questions. The third aim, which relates to the phylogenomics, is as well as to transcriptomics and the analysis of large data sets, is can we use new ways of thinking about network connectivity to discover other genes and possibly even other processes at play in plants, and certainly other genes within the network. So once we start to build a capacity here, we'll start to be able to pick out ways of finding patterns and then attributing some value or importance to those patterns. Okay, so I'm going to switch gears now and talk about the third theme, which is innovating the path to crops. So what I've been talking to you about is thinking about the plant as a whole system. And given that that is indeed the case, we are going to discover that perhaps the best way to improve a crop might be to change the connectivity within the network, or to be able to change multiple components, genetic components of the network at the same time. So Peter Waterhouse will be working on CRISPR-Cas9 and looking at using technology for gene networks to be able to create multiple changes of a network at the same time. And the other aspects that is really important, that not only do we need the capacity, to introduce multiple changes in a network simultaneously, we also need to be aware that the community will need to adopt that technology if we're successful, which means that we need to be making sure that the community is happy with where we're going, but also that the legal frameworks are in place. Without the legal frameworks, it'll be very difficult to get industry to adopt and take on board the technologies. So there's a whole raft of activities in this space that will be led by Brad Sherman. And first of all, thinking about quantitative biology itself. What are the novel aspects in quantitative biology? What is the narrative that biologists use to explain novelty? Because that narrative needs to be captured by law in order to be able to protect intellectual property. What is originality in terms of this space, particularly when you're dealing with extremely large data sets and the novelties actually come out from the way that you've analysed that data rather than the data itself. Another aspect, of course, important to researchers is authorship in this space. And the more we move into multidisciplinary science, the more that becomes important. Now, another important aspect of Brad Sherman's work is the access to genetic resources. This is particularly important for Australia and, well, in any nation indeed, but particularly for Australia with our Indigenous communities and the use of Australian native foods, Australian native plants. There's a number of treaties, conventions and protocols here which are in place, but which need to be integrated and understood. And we need to understand what the fate of historical collections of plants will be in context of this, the access to those plants and what benefit sharing actually means in this context. Then there's legal aspects of this engineering of networks using CRISPR-Cas9 or other technologies, as well as laws around CRISPR-Cas9 quite directly. And another aspect, of course, that probably many of you will be aware of is plant breeders' rights by sequence. Could we be looking at using sequence technology for plant breeders' rights as a mechanism of protecting new varieties? Okay, so I'll just now give you an overview of what I've described so far with a little bit more specificity on actually what we are doing. So we will have a focus in, well, the way I've set up this slide is at the top we have crop and model plants. So we can think of that as human selection. And at the bottom of the slide, I'm talking about diverse plants and ecology, which is natural selection. And we can look at developmental physiology or ecophysiology or the combination of the two when we study different processes. So the focus of the centre in discussions with the CIs is, you know, we all see these areas, day lengths, and how plants are perhaps going to have to be grown under new conditions with climate change. Seed size and seed numbers are plastic traits that the centre could address, and tillering, which is, you know, how many branches plants have, resource allocation as well. These are a major, quite difficult parts of plant success, which clearly involve major networks and multiple networks. At the same time, the strategy that plants use to manage wet or dry conditions, heat, high and low nutrient and light. And we may address this through looking at genes and gene function all the way through to sub and whole plant, depending on the perspective. But the real strength of the centre is in the capacity that we will build in integrating networks for modelling, and then using adaptation theory for predicting network changes, and then the gene technology and legal aspects. And the purpose, because all of this is actually designed with the benefit in mind, which I should have probably presented at the beginning. So the purpose is to improve models and breeding pipelines and to improve phylogenetic diversity analysis. So it's about using prior knowledge to enhance breeding pipelines and to enhance phylogenomic analyses. If we can nail this, and I think we really can, then we'll be producing knowledge on how reproductive strategy and resource strategy are integrated, including the genome environment phenotype sort of scales. This should deliver a step change in the breeding pipeline, and it will certainly enhance the use of knowledge across the genotype environment phenotype relationship across plant species and scientific domains. So I'm talking about the different perspectives of plant science here. And then we should lead to improved use of plants and plant rights. So all of this is really dependent on the maths colleagues amongst the group, where we'll be building this new capacity and then utilising it to improve understanding and prediction. And a focus is really, in a nutshell, about greatly enhancing our use of prior knowledge, so that we're not reinventing the wheel all the time. We're not putting our prior knowledge aside when we analyse a big data set. How do we actually use prior knowledge to its greatest capacity when we analyse a new data set, when we make predictions, when we try to breed, when we try to understand diversity. Now, one of the great things about Centre of Excellence is the ability to train, to foster, to enable, to excite, to build capacity within to the early career researchers, because they're the ones who are going to make the most of this knowledge and this new capacity, this new way of thinking. And Daniel Ortiz-Barrientos will be leading a researcher development working group, and we will, we've already worked really hard on a centre charter, which will enable us to have, really respectful meetings and be inclusive. We're very strong on this aspect. We have quite a lot of thinking about a Kickstart program, which is about reaching out to other researchers to link into the centre, particularly linking through the early career researchers. A travelling scholars program, which will enable researchers to sort of interact with other labs than their own. There's a carer support scheme, which will enable people to attend meetings, and Masters in Quantitative Biology coming out of the School of Biological Sciences at UQ is about, well, the centre will be able to help with placements for those students. We also are working on an outreach program collaborating with the Stephanie Alexander's Kitchen Garden. This is just one of the major collaborations that we are planning to develop, but there's 2,000 schools which are at reach within this program, and they're really interested in describing to children the science that is underpinning food, basically. So how does new technology actually work in agriculture to deliver outcomes to food? How is food actually landing up on your table? So they are very interested in the science behind all that, and so we can help with that, and we can link anyone who's interested in talking with them directly to them. So just summarising then the whole objectives of the centre. As I've been talking about, we're really about integrating mechanisms, linking networks to each other, not just gene networks, but networks that, for example, leaf affects growth of the apex. We're very strong about this integration point because we understand that the lack of integration is really limiting the ability to understand plants as whole systems, and we need to address that. And to do that, we were developing models that connect mechanisms and genomes for predictions. And finally, we're innovating the path to crops, including gene technology of networks, law about networks, and just generally the modern day needs of plant science in relation to law and gene technology. So I'd just like to finish off with some acknowledgements. I'd like to thank our research partners. I'd like to thank each and every lab member of all the labs of all of the CIs, because they have been all very supportive of the CIs while they've been busy working on this research, which has taken a long time to come together with, not yet having spent any money on any research yet. We haven't actually established, but we will be establishing, hopefully. The Vice-Chancellor will be signing off, hopefully very shortly, on the final agreement. We'd also like to thank professional staff that have supported us along the way, including the UQ Research Office and close to home, Claire Fuller and Melissa McCain, who's been working with us for a long time, and the UQ leadership. And with that, I'll thank you all for listening.
Thanks very much, Christine. I think that it's sort of given a really nice introduction to the Centre of Excellence, and now we sort of enter the period where those that are attending the online seminar have the chance to ask you some questions directly. So I'm sure there'll be a number that turn up. There's one from Craig Hardner that I see that I'll read out and you can comment on. Craig's asking, do you see there are opportunities for the centre to link with plant breeding undergraduate and postgraduate education at UQ and more widely beyond UQ?
Yeah, so we have the Masters in Quantitative Biology program, but I think absolutely. I think that the plant breeding is really not front and centre of the biology delivery at UQ, and I suspect possibly across many universities in Australia. So I think that's something that we do need to take on board as a centre of excellence in this space. We need to think about how to enable others to deliver that sort of teaching.
Okay.
Or deliver it ourselves.
Yeah, thanks for that. Second question that came up from Shweta Singh, that really is more about the opportunity to connect with the early career research working group within the Centre of Excellence. And I guess there'll be opportunities through connections through the website for the centre and through other contacts where people can reach out and learn more about the early career researcher. Do you want to comment a bit on that, Christine?
Yeah, so we'll form that working group probably in about February, and we will be coming up with the working group itself will identify some priorities, and yeah, they would be looking for people to participate in the working group. But I think probably the best way for people locally at UQ and at other nodes will be the actual activities that we will run. We hope to be running really useful activities for early career researchers, whether that be connecting to other researchers, whether it be workshops on learning techniques. Really, we want the group to identify what those priorities might be. So you'll be able to contact the group to ignite thoughts and discussions on those things.
Okay. A question from Jeff Norton. Sort of starts off as a bit of a statement, but I'll read it out as it is. No mention of the changes we might expect with changes in pests and diseases associated with climate change. Where does this fit in the analysis modelling?
Yeah, sorry, Jeff, there's actually no mention of pests and diseases at this point in time, just simply through the need of focus. So it's very much a phenotype, phenology, not thinking about pests and diseases. The only interaction we've really got there is through the symbiosis angle, which is obviously not pests, but rather a positive interaction.
There's another one that's an anonymous attendee that's basically asking for some comments about the scope within the Centre of Excellence for short-term visiting researchers, PhD students, and thinking about that from the perspective of will PhD students be able, from outside, to be able to reach out and connect with the Centre of Excellence?
Yeah, we really hope. We have allocated quite some funding for this sort of activity, but I can imagine it'll dry up reasonably quickly. What we have done is made sure that we've got a lot of hot desks within the location within the Goddard building that we've been allocated. So plus we have a very welcoming approach. We've worked hard on thinking about how to protect intellectual property of partners, so it shouldn't be a difficult process to onboard visitors. So we really hope to be able to attract and enable other researchers. I said, you know, I'm very serious about the centre enabling success in others, and helping students is certainly part of that. Depending on the alignment of the goals of that person with the goals of the centre.
Okay, thank you. So those are the questions that have been posted, but it sort of now frees up because there's a few minutes left for me to ask two questions that I've sort of got in mind that really are intended to promote a bit of discussion. I think given you're a very experienced and very well recognized researcher, and you already knew a lot of the people you were connecting with as you're putting this together, what was the most surprising thing from your perspective over the whole journey from the concept through to actually successfully getting your bid up and running? I'm sure there'll be a lot of people that are interacting and thinking about these types of levels of project that would really benefit from your thoughts on that.
Most surprising thing. That's a good question. I think, I suppose a lot of people have said to me, you know, how difficult it is to get a bunch of academics to work together. You know, it's like herding cats. People often say that. And I think actually I haven't had that experience. And I think that's because, you know, we had conversations with people about what their vision was before we sort of started. You know, what do you think we should be doing in this space? Do you think this is a good idea? And, you know, if they thought it was a good idea and they thought that it was a space that we should be in, then it's sort of all been about just working hard together to try and figure out what the answers should be. So I've found that very pleasant experience working with researchers. That it hasn't been personally difficult at all. So that's been great. So I really encourage people to go through this journey from that point of view. I think, you know, provided you're a little bit clear about what your vision is, and that you haven't got all the answers, and that the goal of the CIs is to help you come up with the answers, then you know, that was certainly very good. Working with Mark Cooper was a surprise. I hadn't met him before. Turns out he's quite a good fellow to work with. Yeah.
I'm not going to give you any points for that answer at the end. I've still got my last question. So again, I'd sort of, just to get more of your personal take on this, for you sort of really spearheading this whole effort, what's the most enjoyable and sort of exciting aspect of the centre from your perspective?
Well, definitely it would be thinking about the ECRs and what their futures would be if we can, you know, with a successful centre. I mean, I love being part of the Centre of Excellence I was in before, and it really enabled me to think nationally, internationally. You know, I was able to learn a whole heap of new skills that I wouldn't have had, you know, within my own lab. So I think that's one key part, but also, you know, enabling people to have fresh conversations about where should we be going, big picture. Why are we even doing this? What are the most important things we should be doing in science? And having early career researchers being involved in those discussions with more experienced people at scale and regularly is just, I think, an amazing opportunity. So, I mean, I felt I experienced that, and I'm just really looking forward to that for them. I mean, I think it's one of the most fun parts of our job, isn't it? I mean, you know.
Okay. Thanks very much, and sort of thanks for your willingness to share some of your personal thoughts on the centre. So I think that there are no more questions. You've done a really, I think, nice job of explaining a very large and comprehensive project in the time that's available. And thanks for taking the time to respond to the questions. And congratulations on everything that you've done to get to this point, Christine. And I'm sure now that you've introduced to the broader QAAFI audience a broader understanding of the centre, I'm sure a lot of people will want to follow up with you and some of the other CIs that are actually involved that you introduced. So on that note, again, thank you very much. And I'll close the meeting, and I'll hand it over back to Carolyn. You can finish off the recording and so forth if that's the appropriate thing to do now. Thank you very much.
Thanks, Mark. Bye.